Let us all welcome Dr. Diva Cano to talk on updates on EHP transmission route in shrimp and recommendations for its control in the farm. Diva, the floor is yours.
Okay, I think, everybody can hear me now?
Yes.
Okay, great. Hi, this is me. Thank you for the invitation to NACA, Professor Huang and Dr. Ed. It's very nice to be able to share a little bit of what we have done here, and I might circle back a little bit to the comments that were made yesterday. So I will start now sharing my screen. Okay, so I will update a little bit on what we have been doing in the lab, as well as the work that we have done, so we can understand a little bit where we're coming from and the things we know, things we don't know, and where we would like to go. A lot of this work has been done in cooperation and collaboration with other teams. So as a starting point, as you remember, yesterday it was mentioned that a little bit of the story of this pathogen. So this work has been done in collaboration with CIFAS. So if you remember, this pathogen was found in 2004, but it wasn't characterized until 2009. This characterization allowed for correct diagnostic by histopathological and molecular techniques. It was the first time that we could fully understand and know what this pathogen was. And later on, these molecular and diagnostics techniques were improved by developing highly specific methods that would not cross-react with other Microsporidian species. As you remember, Dr. Kalaya, she talked yesterday about the SWP primers, which are very, very sensitive and very, very specific for this species, and they are suitable for the use in monitoring the samples from animal environmental sources. Is the method we mostly recommend and we mostly use in the lab. It's also available online. This was also made in collaboration with other teams in the UK. However, what happened later on, in order for us to keep understanding this problem, if you remember, in 2012, there was a big problem with the EMS in Thailand. At that time, we didn't know what was the pathogen causing. So this epidemiological study was carried out in 200 ponds across the country to understand this EMS. However, something a little bit unexpected came up. Around that time, we already knew about this microsporidian, but what was not expected was that the prevalence rate of this infection in the country. So around 60% of the studied ponds were positive for this pathogen. And at that point, it became evident that it was a big problem in the industry that has not been acknowledged until then. Like, people did not realize it was going to be a big problem because, as you know, EHP by itself, it doesn't cause mortality, but it's related to slow growth and maybe some other secondary diseases. But because at that point, they were focusing mostly on these mortality syndromes and APEN, EHP was not really in the mind, was not really thought of a big problem until then. So once we understand, okay, this is going to be a big problem, how can we work on this? How can we study? How does it transmit? What are the sources? Where it's coming from? So several models have been developed in the lab to understand, this interaction between the microsporidian and the host. Some of them have been published. for example, the horizontal transmission models by the team at Centex and another transmission models by the teams in Arizona. These models help us understand like the horizontal transmission is the main route of transmission in the shrimp. Vertical transmission between parents to larvae or like it's not really possible. It has not been found in the gonads or the ovaries of the broodstock. So we focus mostly on the horizontal transmission, and this lead to an advance in the understanding on how this process goes on in the field and in the pond, right? There is also a review of the models that are used currently in the laboratories that was also done by the Arizona University. So once we understand the transmission route, that the horizontal is one of the main routes, what happens during the horizontal transmission, or what is it that causes the infection? So I focused during my PhD on the isolation of viable mature EHP spores. These allow to study the pathogen itself and the effectivity assays, and were used to develop and assess strategic tools to control and minimize the spread of this disease. A lot of the work has been done in vitro. So if you think about it in a way, the horizontal transmission, the spores are present in the feces of the infected shrimp, and the feces will go into the water, and then it will go in the pond in the system, either the sediment or any other carriers. So at this point, we were focusing on the mature spore. It was like our key point. And this method is also available online. If you have any question, please let me know. So we have been using this bioassay to study a lot of the life cycle, like how to interrupt this mature life cycle, because if the mature spore is the one that can infect the shrimp, the one that we know so far that can infect the shrimp. So what happened later on, there are some other problems in the industry. Like, okay, we know that is mainly the oral route or the environmental route that plays a big role in the infection. So a lot of people were concerned on when it comes to the feeding practices. So this problem came up with the company that their food was coming positive for EHP by PCR, which can be a problem when it comes to trading. So we had to understand, is it really possible that if the processed feed can or cannot cause EHP infection? We already know from the previous work with the mature spores that they were very sensitive to several conditions, right? So after developing this project, which is also available online, we were able to prove that the pellets or the processed feed that has been subjected to this manufacturing process where high temperatures are used, they present no risk of infection for the shrimp. Even though the product can come out PCR positive, it doesn't equal to infectivity. So this is also available online. Let me know if you want a copy, I can send it to you. So this is part of the infection routes. Now we know like the oral route when it comes to processed feed is not possible. A lot of work also has been done when it comes to making this review it was published a few months ago. If you want to have more details about the EHP itself, it comes with the history of the EHP shrimp, the impact of this pathogen in the aquaculture, biology of the spore itself, which includes the morphology, life cycle, and transmission. I'm not going to go into these details as they are already available, so please let me know also if you want a copy for this. So after we address a little bit of what we have done, what are we doing now? We are working on alternative transmission routes. We know that, as I say, the horizontal transmission by cohabitation, by feeding, they can be possible. However, what happens if you use water that is contaminated? You don't have EHP shrimp anymore in the water, but the water has active spores. As you can see in the pictures, these were isolated from the water column of the tank where EHP shrimp had been kept. So is it possible that if you use the water untreated, can your naive shrimp get infected? So far, what we have found in the lab is that, yes, the untreated water can become a source of infection in PL12. We still have some ongoing experiments to understand and develop the strategic treatment that can be potentially applied in the field to prevent and minimize this infection risk. The manuscript is currently on the works. So as soon as it's available, we want to make this information public. Sorry. And another aspect when it comes to the pond transmission, this is a work carried out by another colleague. She is also a postdoc here at Biotech, Dr. Keek, Dr. Natini, but call her Dr. Keek. So she was focusing on identifying the potential carriers or reservoirs for EHP in the culture ponds. For example, bivalves, zooplankton, or other crustaceans. And she identified species like a genus of mussel called Mytilopsis, which is commonly found in the ponds, and it can be a potential carrier. I think her manuscript is almost finished, so please look forward to it. It will be very, very interesting. So the recommendation for this is in your pond, it's better to get rid of all the potential carriers as much as you can and to remove them. And in the past, the work that has been done in vitro, we tested several chemicals and physical treatments with the mature spores to see which one had a potential to be used in the field. The ones that showed the most potential were potassium permanganate, chlorine, and formalin is very popular, but it did not have the same effectiveness as the other chemical treatments and ethanol. And when it comes to physical and parameters, the temperature, high temperatures, 75 degrees or minus 20 degrees can inactivate the EHP spores. So if we inactivate the mature EHP spores, it means that the infection process will not be able to be carried out. And another aspect we found is a high pH are able to trigger this extrusion of the polar tube. So if you trigger the extrusion of the polar tube before it gets in contact with the cell, you will interrupt the infection process, because the main role of the polar tube is to go through the membrane. So if it's not able to, it will not be able to eject the infectious substance. So how was it applied, this knowledge that was generated? I would like to mention to you a little bit about the project between the local farmers of where we collaborate with them in order to apply and develop techniques that can be used in the field and in the farms. So there was a problem with one of the soil ponds that they were positive for EHP, the samples. So during the treatment between crops, the pond was sampled in the sediment line. There were 4 different points. As you can see, 3 of those points were heavily positive, like very loaded with the EHP spores. So the soil that was sampled, right, was very contaminated. So they were treated, this pond was treated with lime, calcium, magnesium oxide for 1 week. After 1 week, the same points were sampled and the infection level was revised again by the SWP method. And as you can see, after 7 days of treatment with the lime, the infection level is almost non-existent, like it reduced. So it means that the recommendations that had been done in the past, they do work in the field. However, a lot of more work needs to be done in understanding how can we really apply this knowledge in the working field, right? And even though a lot of information has been generated, not just by us, but by other teams too, which is great, there are still a lot of things that need to be addressed. There is a lot of information that we still don't know about EHP. We don't know, is there any other life stages in other host species? So far, we have found that the EHP replicates in the host shrimp. However, can it be possible that it can replicate in other host species, and which host? Is it this life cycle, is it the same? So a lot of work needs to be done in that aspect. Are there other alternative routes of transmission in the shrimp? For example, feeding batch exposure, maybe somebody can come up with a model that we haven't really thought about it, like the transmission can happen in a certain way. We don't know if there is any different types of spores for different types of infection. For example, some authors, they say that Microsporidian spores, they have different, how I say it, they have different work, let's say. Some spores are specific for cell-to-cell transmission within the host, and some spores are for transmission, like in a way that they go into the environment to find a new host. So could that be possible? That has been seen in other species, but here we don't know in EHP. We don't know if there is any other ways to trigger the spore or to inactivate the spore. So far, we have seen chemical and physical parameters that can be used. However, we don't know, like radiation or any other process that can interfere in this infection process. And are therapeutic methods possible? These could be seen first in the laboratory model by using shrimps that are already infected and see if the shrimp that is already infected can decrease the infection level or can recover from this infection. We still don't know if that is possible. So as Dr. Tim said yesterday, we hope to know what other people are working on and see if there is any potential collaboration or like to open a communication channel, see what you guys are doing. Do you guys have any information about these? That would be very, very interesting to know. On my side, that's about it, and thank you for your attention.
Thank you, Diva. A very nice presentation, especially on the management and prevention at the farm level. So there's one question here. Can you explain the mode of action of the lime or the calcium magnesium oxide that you use in the pond against its action against EHP, maybe aside from raising the pH of the soil or the water?
Yeah, so this was used, like I said, was used in the farm. There were two lead scientists involved in the process. Uh, the way they use the calcium oxide is the lime is this lime like oh, what was the correct name? I'm sorry, I forgot the name, but it's a type of lime. And it's this powder, they put it in the pond. I cannot tell you exactly how many kilograms because it depends on your pond, right? And the characteristics of it. So the way they use it is they spread a powder in the pond, enough powder to cover the area, and they add water around 10 centimeters. So the way it works is that they raise the pH up to 11, at least to 11, to make sure like you have to make sure like all the bottom is covered and that the pH is high. And then they let it for 1 week. If I'm not sure if does it degrade the spore wall over time or the spores over time, I cannot tell you that. However, after taking samples from the same spot, you will see the level of detected by PCR, the level of detection was very, very little. But you have to remind that the spore, they need a host or a reservoir in order to survive because they cannot replicate in the soil by itself or in the water by itself. But if there is some kind of reservoir that you have to clean it or disinfect it, that would be the best course of action.
Okay, thanks, Diva. There's another question here. What is the best life stage to detect EHP with PCR? I'm not sure if it's the life stage of the shrimp or the life stage of the parasite.
Okay, for if you talk about shrimp, for example, if you use PL, you just remove, you use the cephalothorax and you remove the eye stalk. That is the best way. If the shrimp is a little bit bigger, let's say like a juvenile, is the hepatopancreas. We use hepatopancreas. However, when it comes to the life stage detection, this SWP primer is based on a spore wall protein, which is expressed in every stage of the development of the shrimp, or usually when there is when it's becoming mature, or maybe a little bit more. So it doesn't really, when you use the whole tissue, let's say, you will have many life stages of the spore. You will have mature, you will have I say the plasmodial stage, you will have sporons. So it's like a combination of stages. So if there are mature spores, you will detect them with the PCR.
Okay, how about the chlorine as a potential disinfectant? Can you elaborate on what is the action of chlorine?
That is part of the ongoing work. We have tested in vitro. It's in the bioassay paper. There in the bioassay paper of where we explain how to purify the active spores. We did in vitro testing where we use the purified mature spores that were active, and we expose them to different concentrations of chlorine for 15 minutes and 24 hours. we have the results that it worked in vitro are in the paper. However, when it comes to the field, that is the situation. Like, you have to adapt what you see in the lab and in the research to your conditions in your farm, because the conditions changes. They're very particular to the type of farm, to the design of the farm. So when it comes to chlorine, what we are doing is that we are testing if it can help with it. It is recommended, but we are seeing if it really can stop the infection process.
Yeah, based on your studies, how long is the life cycle of EHP? Do you have any information from the results of your studies?
What we have read about other species, the spore can complete its life cycle in a matter of days, meaning like from infection, plasmodium to mature spore. However, once you purify the mature spore, it remains active. From what I have seen here in the lab, I can only keep it active around 1 week, because as we say, they need a host to obtain the energy. If you remember yesterday, like that it was mentioned that they obtain the ATP from the host. So once you remove them from the host, there is no way for them to obtain that energy. So after a period of time, the activity will decrease.
Okay. If Professor Suba Basu is listening, I cannot get your question. Can you unmute and ask your question directly? Professor Suba from
Hi,
Malaysia.
I don't know.
Thank you.
My question is, EHP has been present for many decades. Why is it now shrimps are susceptible to it? So why? They used to be very tolerant, but now they are susceptible. Or maybe you could shed a light to this.
I think that question has many. The answer is not an easy answer for that, because as you remember, this microsporidium was seen in before, but it was not characterized, and because it did not represent a problem, the attention was not focused on that. It was later on that people started looking more into it. Like they say, how can you find it if you're not really looking for it, in a way? So the spread might have been maybe some leniency when it comes to biosecurity, and maybe we got a little bit too comfortable when it comes to enforcing biosecurity issues. However, it has been reported in many papers, and I think it's doable that even if you have EHP infection, you are still able to culture your shrimp to a profitable, marketable size, as long as you keep good culture practices. The important part is also to be very careful when it comes to hatcheries, because that is like the start of the culture cycle, let's say. So if you're careful from the start, then you should not have a problem. It is now like a global issue. It's not like a regional issue anymore. It's not just one country having the problem, but it's many, many countries. So I think in order to be able to answer that, we have to collaborate and understand this pathogen better, and why did it happen? Why did we reach this point? And how can we deal with it? Either overcome it or learn to live with it.